Welcome back, friends. In this video we are going to see about the genetic code. So in the last session we have discussed about transcription. So if you have not seen that video, you can see the description. I have given the link, you can see the video. What about transcription? So transcription means the conversion of DNA into RNA. The message which is stored in the form of DNA will be transferred into the messages of RNA. That means the nucleotide bases, the sequences will be converted into RNA sequences. So these RNA sequences will be responsible for the production of proteins. So what is genetic coding here? If you see, means the sequence which is present in the RNA. So that denotes genetic code. So all these bases which are present in RNA, they together, like they will be separated into three bases. Those three bases denotes each code. It is codon. So if you see here the DNA, so DNA consists of two strand double helical structure, that is double stranded DNA will be there. So imagine this as a, like for example, imagine this is the sequence of the DNA. So from the DNA, RNA will be produced with the help of process called as transcription. So this is the sequence of RNA here. So here mRNA, that is messenger RNA, carries message from the DNA. So here you can see 3, 3, 3. Like that you can split the bases into three bases. When the protein synthesis taking place, during that time the enzyme will be reading in this direction only. While reading, it will be reading as three bases, and based on that codes only the amino acids will be added to the newly producing proteins. For example, here you can see A will be single nucleotide base, and this will be a single nucleotide base, and this will be the single nucleotide base. Totally 3 bases together. This is called as codon. So codon means the code which is present in the mRNA. That is called as codon, which codes for the amino acid. So here 1, 2 and 3. Here in this case 3 codons are present. So here in this case the main features of this genetic code is always it will be in triplet format. By triplet means only 3 bases, not more than that. If you consider as 4, it will not code for any amino acid. So based on the experiments and research, they found out that only the 3 bases can produce amino acid, not 4 bases cannot produce any amino acid. So three bases together produces the amino acid. So this is a single base, like single codon. So totally many codons will be there. So what are the main features? If you see, the first will be triplet, always will be in 3, like that only it will be there, and it will be like contagious, like contiguous. What is contiguous? So contiguous means continuous format. Between the bases there will not be any comma, like this slash, semicolon, it will not be like that. Like this comma, it will not be there. Continuously the bases will be present, but the enzyme has the capacity to read the bases, three bases. There will not be any commas, like semicolon, nothing will be there. That is called as contiguous. And continuous form only the bases will be present in the RNA. So genetic codons will be continuously present, but it will be read as three bases. That is contiguous. And the next thing is degenerate. What is degenerate? Degeneracy means, for example, before going to degeneracy, you should know about this unambiguous. Unambiguous means specific. So each codon is specific for each amino acid. It will not code for any other amino acid. For example, if you take AUG, this will be always coding for methionine amino acid. Wherever from the bacteria to the higher organisms like human beings, if you take AUG, always this AUG will be coding for the amino acid called as methionine only. It will not code for any other amino acid. That is called as specificity. That specificity is called as unambiguous. So and he said, right, from bacteria to higher animals. So this base will be common. So it is called as universal. Universally from bacteria to higher animals, this will not change. Suppose the AUG is present in bacteria, that will code for same methionine. If it is present in tiger or cat or lion, there also it will code for methionine. Humans also it will code for methionine. In plant also it will code for methionine only. That is called as universal. So what do we unambiguous? Specific. Always this code means this amino acid only, it will not change. Okay, now we can see degeneracy. What by degeneracy means, like here one codon, one amino acid. Degeneracy means this same amino acid can be produced by some other codons. For example, if you take UUU and UUC, these two are UUU, UUC. These two codons will be coding for the amino acid called as phenylalanine. Phenylalanine. Okay, this phenylalanine amino acid can be produced by two different codons. Okay, but it will be specific. So this codon also produces phenylalanine. This codon also produces phenylalanine. That is called as degeneracy. Whereas the same codon cannot produce for any other amino acid. That is called as unambiguous. So for better understanding, imagine director and hero. So suppose the director is making some big project, okay, big budget film they are making, means single director will be busy with making the single film only. Okay, so imagine that as specific, okay, director. Director means in this case imagine the codon. Whereas hero, the same hero can act in 2-3 different directors within the same same time, same duration. Like same duration means within the same month 2-3 films will be released for the same hero, right? So during that time he will be acting in 2 different directors. So imagine this amino acid as heroes. This hero can be acted up with this director also. This hero can be acted up with the another director also. So that is called as degeneracy. Simply one amino acid is specific for one codon. One codon will always produce for one amino acid. That is called as what? Specificity. The same amino acid can be produced by some other codon also. That is called as what? Like degeneracy. Degeneracy means different. Same amino acid, you can see same amino acid, different codons. Degeneracy. Unambiguous means this codon will always produce phenylalanine only, to not produce any other amino acid. That is called as unambiguous. You should not confuse with this. Definitely the NEET question will be coming based on this. Okay. So next, if you come in here, another important thing you have to remember is AUG. AUG is the start codon in all organisms. So whenever the protein is synthesized, the first codon will be AUG. A is the start codon. It is the initiator codon. So in some rare cases in prokaryotic organisms, rarely you can find this GUG that codes for valine. This valine can also act as initiator amino acid, initiator codon in some rare cases in prokaryotes. In maximum cases, AUG is the start codon. And the next thing is, these three are called as stop codons. So before going on to that, you have to remember this thing, this table, how they are making these codons. Okay, totally 64 codons are there. 64 codons you can see. Here it will be 4, here 4 and here. What are these 4 things means? These are the bases which is present in the RNA. RNA consists of 4 bases as you already know. Adenine, then what will be having? Guanine, uracil and cytosine will be there. So these 3-4 bases only I have mentioned here. So this will be coming in the first position. For example, first position if you are mentioning U, then this case, okay, U will be the first position here. And second position will be U, again U. So the third position you can see here again U will be there. So U will be one codon. You can take next here example. First position will be U, second position will be, for example, here we take A, and third position will become U. Again UAC. UAA, UAG will come. Here also you can write, then you can write U, second position will be U, and third position will be C. Again U, and here what is that? A. Again UUG. Next UCU, UCC, UCA, UCG. Similarly, for all the things you will be getting totally 64 codons. In the 64 codons, only 61 codons will be coding for the amino acids. Remaining these 3 codons will not code for any amino acid. So only they are called as stop codons. Whenever the mRNA is going for protein synthesis, whenever there is a stop codon, the protein synthesis process will be stopped. The protein will be coming out from the process. That is called as stop codon. They will not code for any amino acid. And if you look at here, 61 will be coding for amino. How many amino acids they will code means totally 20 amino acids they will be coding for. So from this you can easily match here. Okay. So codons are more in numbers, whereas like amino acids is very less in number. Okay. So 61 codons is coding for 20 amino acids only. That means many codons can code for single amino acid, whereas Single codon codes for only particular amino acid, specific. You should not confuse with that. So that is called as this unambiguous degeneracy. So this is the overall concept of this genetic code. Why we need to need this code means if there is any change in this code, it leads to mutation, genetic disorders, many problems will be there. So based on these codes only, amino acids will be produced. These amino acids will be joined together to form protein molecule. So this is all about the genetic code. In the next session, we will be discussing about protein synthesis in detail. Thank you friends. Thank you for watching this video.